Cool. All right, welcome. Good morning, everyone. So today it's going to be a one-man show, pretty much me. So what I was specifically want to talk about is how to write a good proposal. This has gotten to be significantly more important than ever, considering the current funding landscape globally. And hopefully, as of yesterday, you have seen the global community trying to help, coming up with all sorts of initiatives, all sorts of programs to allow scientists in under-resourced regions to gain access to a lot of the programs, funding, as well as microscope instrument all over the world. And unfortunately, over the past 2 or 3 years, the quality of the proposal coming from various regions, especially Africa, has been pretty poor. Poor to the point that some of the programs have completely canceled their outreach to Africa, and we have to solve this problem. And so today, you're going to hear a lot of unvarnished truth, and hopefully you will stay with me, because as difficult it is to hear about what I'm going to say today, it is actually extremely important that we fix this problem, so that we continue to be able to take advantage of what the world is trying to share with us, and so on and so forth. And you have heard from Denise yesterday that even for imaging for all, the quality of proposal has been, to some extent, pretty shocking to some of the programs. And the AIC, for example, has also been the receiving end of that. And even infrastructure within Africa, such as the Africa Microscopy Initiative, Imaging Center, or the Army Imaging Center, we have faced similar problems. So what I'm going to go through today is basically not only how to write a good proposal, but more importantly, and what I'm hoping to be able to do is basically how not to submit an irrelevant proposal. And I've given this talk to a large extent online for ABIC, and I would like to maybe regurgitate some of the points that I've made earlier and maybe add a few more things. Okay, so I'm going to give you a few case studies, and I think this is important. So this started out with the ABIC SWIFT award, right? So this is a transcontinental mobility award that ABIC created to basically support travel for African researchers to carry out imaging research within the continent. And the goal here is that this is to support international travel within Africa for the purpose of carrying out microscopy experiment. And even with that, very, very simple, very, very straightforward call for application to support international travel within Africa. This is the kind of applications that we have received. The goal is that let's just see how many people actually follow the application instruction. The instruction says 2-page maximum. 50% of the people cannot follow that simple. So that is 50% right there. Administratively rejected. Boom. Half of the application are gone. Just like that. And you should see previously that this is imaging motivation. You got to do microscopy. It's ABIC. It stands for Africa by Imaging Consortium. Even then, 17% of people don't even know that that is the imaging motivation. And 50% of that is semi-okay or poor. And again, this SWIFT award is to support travel. And 50% of the applicants do not understand that this motivation of this award is to support travel. And they ask for research funding, they ask for this, they ask for that, they ask for staff, they ask for salary. Again. So you can now see applications broadly left, right, and center off the radar very, very quickly. International travel versus domestic. 69% of them understand that it is international travel within Africa. 1/3 of the applicants don't even realize that this is to support travel within Africa. And they were like, can I go to my university next door or in a different city? And again, people just don't read. And this is the kind of challenges facing a lot of the African applicants. And you will see more and more of that. And the appropriate scope of work, I suppose that this is just for a few weeks. 58% of the applicants don't understand what is the scope of work. Some people actually suggest works that are, meant for years of research. CAT info necessary to request it. 75% of them don't. It has to be imaging work related only. 33% of them say no. Basically, that's another source of the problem. And then case study number 2. Now, this is to visit the Army Imaging Center at the University of Cape Town. So the Army visiting program is basically to support, again, travel and microscopy work to the University of Cape Town Army Imaging Center. And we have 5 instruments at the Army Imaging Center for up to 4 weeks. So you can apply to go there and do works that requires one of the 5 microscopes for up to 4 weeks, right? And the access will include dedicated assistance from people like Mike and Karen, so on and so forth, some guidance, training from a technical expert who will work with you to perform successful and informative experiments, right? And it is free to use. You can support up to 2 researchers from each selected project, so on and so forth. We encourage all interested researchers planning application for the next open call to talk to the Army staff. So this is the breakdown of the applications that we have received or Mike has received. 12 of them did not read the instruction. 4 of them wrote a proposal that is not understandable. Scale too ambitious. Scope exceeded microscopy. People started to do genomic studies, mass spec, flow cytometry, whatever that you can think of, or no clear biological aims. Not feasible at the Army Imaging Center. The use of microscopy is not rationalized, and so on and so forth. And you'll always notice that the inappropriate application coming out of Africa, unfortunately, always never fail. That's the whole problem we're trying to solve here. It overwhelms well-formulated application time and time again. Then case study number 3. This is the AIC at Genelia, which you have heard yesterday. You know about the application. The instruction is very, very clear. You have to have an abstract, specific aims, a little bit of preliminary data, justification for the use of AIC instrument, a little bit of experimental design, and data quantification strategy, measurable outcome, right? Now this is becoming worse. You are now competing with the rest of the world. This is no longer just an open access to African scientists. You're fighting against the whole world now. And we talk about how we're going to review. We talk about the review criteria, what kind of personnel we hope to be able to bring in. No need to ask for budget because we provide on-site housing and all that stuff, right? So we didn't get that many African proposals, understandably. It's very competitive and more higher end instrument. 2 out of the 4 African proposals did not contain even a project. They fill out the form and send it in. What are you coming to AIC to do? And we open up, wait, did we miss something? We actually emailed the applicant. Did you forget to attach something? No. So that's bad. That's very bad, right? And I can show you some of the randomly provided information. So this is a project, Advanced Imagine. I don't know what that means. So First of all, that's really bad. And which microscope you like to use? They selected two instruments. Very difficult to link those two instruments together. One is the focus beam SEM, the other one is the lattice light sheet microscope. So in the form, we always ask for what is your specimen? Give us a little bit. For example, are you working with Drosophila or zebrafish or Gila cells or something? They thought it was a multiple choice. So they selected the Drosophila. List all the recombinant DNA to be used along with your transaction and transaction methods, because we have to deal with safety issues and everything. Again, we give example, live act, GFP, transient transfection, and lipofectamine, or whatever. They thought, again, that this is a multiple choice, so they randomly picked one. Fluorophore to be used. Example, again, they thought it was a multiple choice of one of the two, so they picked one. So on and so forth. I have no idea what advanced imagine is. So on and so forth. I think we covered that. And our microscopes, we talked about all this. And we basically, in the website, actually almost every page of the website, we say we would like to help. Please talk to us. Please talk to us. Nobody did. And 1 out of 4 African proposal requests to use a microscope that is not on the AIC instrument list. And then here's not even just to use microscope, right? So all the case studies that I've covered up to this point about going to another place to use microscope, whether within Africa or on a global stage. And last year in May, we run this conference at Engineering. And the title of the conference is Microscopy Technology Dissemination to Underserved Communities. And I was one of the organizers, and it's another one, and then Leonel Malacrida from Uruguay was another one. And this is, as the title basically suggests, is for all the global community to come together, especially from under-resourced community, just to figure out how we can together brainstorm what is the best approach to bring more technology to the underserved communities, right? So this is clearly the gist of the application of the topic. And I'm going to show you, with the permission of a couple of the African applicants, to show you what they have written in their abstract. So I'll show you a few, some that are good, some that are bad. So basically, we actually write down a simple synopsis of what this conference covers, right? As microscopy gains acceptance as an indispensable tool in life sciences, the equitable dissemination of this highly resource-intensive technology remains a challenge, right? So on and so forth. There are many individual solutions that has cost-effective hardware, open source software, capacity building, training program. However, there is no concerted strategy to integrate all of this together. And this unique conference will explore the challenges, solutions, and opportunities in microscopy dissemination. It is not meant, and in fact, we purposely discourage people from MIT, from Caltech, and all those places who develop very, very advanced microscope from coming, because this is not the platform. This is not meant as a platform to discuss state-of-the-art imaging technology, but rather aims to bring together developers of easy to disseminate hardware, software, and imaging probes, as well as representatives from a global imaging organization, and basically to talk about strategies. Abstract 1. The first word already killed this application. Autophagy is a cellular process that we break, blah, 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 blah. We're not there to talk about specific scientific topic. We're there to talk about technology dissemination. The very first word killed that application instantly, right? And talk about macrophage and talk about machinery of tuberculosis. So again, you're not customizing your application to whatever that you are trying to apply to. Abstract 2, at a time when advanced microscopy is a key yet inaccessible technology for biomedical research. This presentation highlights the transformative presentation approach of open source microscopy. So basically, they are talking about how a focus on 3D printing can help develop low cost microscopes, so on and so forth. Okay, good. Thank you. So this particular abstract talk about how we can evaluate open source hardware and why is 3D printing our 3D printed microscope is important. And then abstract number 3, spinal cord injury. Again, first 3 words kill that application instantly. If you are the one sitting trying to select which application to invite to this particular conference, you would automatically select abstract number 2, right? Because it actually fit that conference. The importance of low cost design for technology dissemination. Abstract 1 and abstract 3 are out. And guess what? Abstract 2 is not from Africa. Abstract 1 and 3 are from Africa. Problem, again, not customizing your application to what you're trying to apply to. So this is the one that got accepted. All right, and I want to highlight a few things that every program, every platform you are applying to will tell you what they are seeking in the application. This unit conference will explore the challenges, solutions, and opportunities in microscopy dissemination. Presentation and discussion will focus on how to integrate common goals and unified strategies to bring imaging technologies to the broader range of community. So this, pay attention to what the program is trying to tell you. It is quite important. And yesterday I talked quite a bit about approaching the organizers or the platform owners or the program officers to talk about, to ask questions. Now, this guy, if I am not mistaken, is from Poland. I could be wrong. And he and I have never met, but somebody from Poland actually sent an email to me. Hi, Dr. Chew, how are you doing? I just saw the software part is part of the conference you are organizing, and I'm always interested in the topic of dissemination. Oh, yes, they built up collaboration in Brazil. This is from Portugal. I remember that now. Specifically to underserved scientific community. I built up collaboration with Brazil in my times and would actually love to somehow get back on track. Do you think I can have an active role? I can talk about, all right, so do you think I can have an active role in this conference? I can talk about our common project, my experience, how I think would be done. But I can take no for, of course. He reached out. He has no idea what to write, but he reached out. And I spoke to the other 2 African candidates who very generously shared their failed or rejected application with all of us, just so all of us can learn together. I asked them, and the funny thing is that both of them know me very, very well. And we talk all the time. This guy has never talked to me. So I asked us to, you guys know me very well, and I'm one of the organizers. How come you don't talk to me? Oh, we don't know. The idea of talking to the organizer or talking to the program officer rarely crossed the mind of African applicants. We have got to change that. We absolutely have got to change that. Talk to people, right? All right, case studies number 5. Now, this is the slides that Denise, who spoke yesterday, shared with me. Everybody know what Imaging for All is now, right? You are going to various places supported by this program to basically do some imaging, and there are many tracks. One of them is an experimental track where you actually go to a place to do experiment. So this particular proposal basically said they want to look at reactive oxygen species and autophagy using flux assay. And then imaging-based assay to monitor cell survival, cell integrity, stress responses over time in different cell culture. They want to perform label-free 3D imaging to assess effects of oxidative stress in different cell types. They are going to perform morphometric analysis to look at the neuronal soma size, axonal dendritic arborization, neurite length,
branching complexity. They want to quantify the synaptic architecture and the synaptic density and connectivity inside the sample. And they want to do real-time imaging of calcium flux, mitochondrial dynamics in some sort of a experimentally challenged cells. And they want to do iPSC differentiation into 3D brain organoid for light sheet microscopy. And they want to do high content screening for 2D neuronal cultures in 96 well plate treated with certain compound. All of that in 3 weeks. All of that in 3 weeks. How is that possible? That is not possible. That scope, again, that is immediately administratively rejected. It will not be sent for review because it is so outlandishly crazy. There's no way you can solve this problem. And so another one, lack of clarity. Sometimes the proposal never specified the dyes, the probes, and the antibody to be used. We're going to look at this. We're going to look at that. What label are you using? Is your label? Do we have the laser to activate your probes? We don't know, right? You never tell us. Not specifying how many compounds to be tested, how many conditions, not describing or thinking about control samples. There is no positive control, no negative control. Underestimating the time, 1 day for TEM sample preparation in the timeline. Even in the most advanced lab, that takes weeks. Ignoring or not understanding optimization needs if the sample preparation workflows have not been established, not considering whether the host facility even have the equipment that they need. You apply to a facility that has no EM and want to do EM work, for example. Insufficient consideration of the inherent variability of biological samples when you're trying to evaluate the differences or trying to interpret results, and weak or non-aligned readouts for addressing the stated aims, right? And then here's another example. So this is just to develop skills. So they have experimental tracks, but they also have skill development track. I want to receive hands-on training on the use of confocal fluorescence microscope and electron microscope. I want to learn image analysis. I want to also learn how to run imaging core facility and training on sample prep in 2 weeks. In 2 weeks. Again, administratively rejected, right? And then AIC runs this 2 weeks microscopy bootcamp. You should pay attention to this because this is actually one of the very, very few microscopy bootcamp in the world that is completely free, completely free. So obviously, this is open to the whole world. And because of the Africa Microscopy Initiative and everything, for the first time in history, the number of proposals coming from the Africa beat out those from Europe. I was very, very happy when I first saw that statistic. It's like, yes, finally. This is really, really encouraging because now African scientists are starting to think that, you know, to really consider that all this international opportunity are available to them, which is pretty much the gist of what we have been trying to do. But again, this is globally competitive. So the fact that we have 23% of the application coming from Africa is a wonderful sign, and I love it. Okay, so 21% of the submitted applications are from Africa, which is really great. Within the first round of applications, 68% of them has been completely wiped out because they are completely off-target application. And this is when it becomes extremely difficult. I'm not speaking to any audience in particular, but I want to make a very, very important message to whoever is listening. Please do not lie on the application. People know. We asked for pixel reassignment. How experienced are you with pixel reassignment microscopy? This is basically ARI-scan, for those of you who know what ARI-scan is. Africa is more experienced than the rest of the world. Structure illumination, super resolution microscope. Africa is more experienced than the rest of the world. Stimulated ablation, this is an extremely expensive, more than 1.5 million a pop of an instrument. Africa and the rest of the world almost matched. there is no stat in the whole continent. Fine, but again, they could be, you know, African diaspora will take it, but that is still a pretty serious statistics. Localization, super resolution microscopy, almost as good as the rest of the world. Expansion microscopy, better than the rest of the world. Light sheet microscopy, how many light sheets are there in Africa? Significantly better than the rest of the world. And there are also people who will basically say, so we ask people, rate your own, I wouldn't call it rate, how often do you use the various different modality of the microscope, and how proficient do you think you are? There are some people who say, I have never used wide field, I have never used confocal, but I'm an expert in super resolution. You know, that you all know that is not possible. Again, if you are not sure, ask. I think this is the guy from Poland, finally. So he wants to apply. I think it's a good opportunity. As one of the course director, I suppose you have a lot of experience in rating app. I wonder whether you can give me a few tips of preparing an application, which I hope I can increase my chances. So again, if you think about this on a global scale, people who are in a much more privileged situation than many scientists in Africa will continue to think about how they can make their application better. On the other hand, which makes matters significantly worse, is people who are in underprivileged condition continue to jeopardize their own application without talking to us. Always talk to us. So let's talk about how to write a good proposal. Submitting an application and proposal is not buying a lottery. Please don't leave it to chance, right? It is really, it is not because if I apply to 50 various places, one of them has got to stick. No, it is not buying a lottery. Also, very, very important, none of this is first come, first serve, right? And we let, like the AIC proposal, for example, we let it circulate for 2 months. The Army Imaging Center, we let it circulate for at least a month, if not 2 to 2.5 months, even longer. We let it circulate to give people a chance to prepare, to give people a chance to talk to us, and to give you a chance to write. It is shocking how fast that within 24 hours, we already got like 30, 40 applications from Africa, as if it is first come, first serve. And that is a problem. Give it some thought, all right? Don't apply if you're ready. And again, people come and talk to us and look at, this is actually from a very, very big university in the United States. I want to thank you for spending so much time with us, providing us with a wealth of information. And they originally want to do a specific technique. And then after talking to us, they realized that that is not the best approach. And they have decided that they want to switch approach per your advice. To do this well, I think we need to first retool how we can get comparable level of blah, 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 blah. I very much appreciate your encouragement. However, we feel a lot more confident when getting a reliable outcome if we can give ourselves a little bit more time. They decide not to apply for that round because they don't think they are ready, right? And the drastic difference in attitude is very, very different. And this is actually a letter from Cornell Medical School. They don't think they're ready. So they take it so seriously. It's like, no, we are not ready. We're not going to apply. Whereas, 24 hours later, quite a few proposals coming from Africa. And we have to take everything, any kind of this opportunity significantly more seriously. Big mistakes. The title of this program sounds awesome. Let's apply. And let's apply within 24 hours. Bad idea. Let's submit as soon as possible. This must be first come, first serve. Let's just submit something and see if I could be lucky enough to get accepted. I have no idea what they're asking for in the application, so I'm going to guess. I don't have time to write the proposal, so I will attach my previous grant. This is where many people exceed the 2-page limitation because they attach the 50-page grant on it. I don't have time to write, so let's just attach it. The bigger the project, the more likely it'll be accepted. That leads to, you know, a 5-year project applying to a 2 weeks program. And I don't want to bother the program officer. They're probably very busy and won't talk to me anyway. As I mentioned yesterday, this is probably the biggest mistake of them all, because the moment you talk to us, you will realize that you don't have to worry about all the first 6 topics. You actually have to worry about your science in general. So that is very important. So good practices is that you read the call for proposal or the call for application. If you have read it, read it again. What does the program on the call for application look to support, right? Ask yourself if you can take advantage of the opportunity. If you're not ready to take the opportunity, don't apply. Are you eligible? What would you do if you're successful? Is your plan aligned with the program, right? By keep submitting the proposal to all this program, you not only jeopardize your own application, you'll jeopardize the success rate for all your fellow African scientists, because now you create this very, very bad bias in the global north, which I'm trying very, very hard to correct. And everywhere I go, I have to fight with people, is that the global south people don't know what they're doing. I'm trying very hard to change that, so please help me. And determine what the evaluation criteria are. Read the proposal again. Read and read and read and read, and contact the program to investigate. Understand what is being asked of you in the application. Do not select all the options, like the one that you saw at the AIC. Do not cut and paste example. Do not pretend you know. Not sure, contact the program. You are sure, contact the program. Just contact the program. Tailor your application to the program. Don't expect the program to cater to you. We can't cater for it because we have a very strict mission of what the money and everything has to be spent. So before you start with anything, read and read and read again, and then think. Gotta think. Think about how the program will benefit you. Do you have the appropriate justification to apply? List all your questions, and then speak to the program officers or the conference organizer. Investigate what they're looking for. You will be surprised how much guidance you get from people when you talk to people. Some of them will actually offer to help a lot more than you think they would. And then strategize. Print out a program. Always print it out. Don't rely on memory. Print it out. Every program, every conference will have some sort of a review criteria. Print it out, and then write your proposal and check it off to make sure that you have addressed every single thing, all right? Because guess what? All those bullet points will be shared by the reviewers. The reviewer are instructed to follow those points and review your proposal. So the very first thing you want to do is to check to make sure that every single point is addressed, so that when the reviewer read it, they can actually score. Don't write your proposal in such a way that the reviewer cannot score, because in the afternoon, we're actually going to turn the table. After you have written your proposal, we're actually going to teach you how to review a proposal. You're going to review each other's proposal. All right. And so now that gets to the last slides, which is the class exercise. So we are going to pretend that we are going to write an application to the Army Imaging Center in Cape Town. And this will be done as a group exercise. So if I'm not mistaken, Precious, that their name tag has a color code, am I right? They don't have name tag? Oh, great. Okay, so you guys know your group. Find your group member. Then within the group, decide on a project that you guys can write, all right? You don't always have the most let the most senior person in the group dictate. They will likely dictate, but find them. So find a project that is matured enough that it can constitute a reasonable proposal, and then read the application instruction, which has been shared, right? And I'm not going to hold your hand through that because I purposely want you guys to read that instruction carefully. And then collectively, keyword here is collectively, don't let one person dominate the group. I want everybody to be involved. Work on the scope, the aims, the experimental design, and the choice of instrument as if you are applying to the Army Imaging Center. And then in the afternoon, we will give you plenty of time. I don't know how many hours. And then in the afternoon, we will learn how to conduct peer review so that you now see when you are at the reviewer's role, how your hands are tied to review only certain things. And if you don't address that, the reviewer cannot score and they will always kill that proposal. So this actually become an even more important exercise than the writing itself.
Okay, so we are going to move on to the review of our proposal. So you are going to share your proposal with 2 other groups, and those groups will, so each group will review 2 other proposals. And then we're going to have a bit of a feedback session. But I wanted to give you some insight into what the review consists of and what you must keep in mind. So this was a real life exercise, a real life example of what we did with the AMI. This rubric that I'm presenting here is a simplification of what the reviewers actually saw. So I just captured some very specific information. But as a reviewer, in the case of this visiting researcher program applications, I sent to them the review scorecards as well as the initial application instructions. So you as a reviewer need to understand what was asked to the applicant, but you also have to understand what are we trying to pull out of the applications, what information is important. So my suggestion in this case, and what I'd hoped all the reviewers did, is that they understand this rubric before they start reviewing proposals, okay? But before understanding this rubric, the reviewers also need to understand what the application consisted of. So they need to look at the instructions that you guys just worked with, okay? So you had various headings that you had to write, so project background, a justification, an experimental plan, I think outcomes was one of them. That doesn't directly translate one-to-one to the review. So in the case of the AMI proposal, we were looking to support research that was either very high impact, had a very strong justification for microscopy based on whatever setting it was. In our case, we were expecting applications from low resource environments that didn't have access to these tools. So those were the 2 key things, to support good research and also to provide an opportunity that wouldn't otherwise exist, okay? In our review, we have to keep that in mind, okay? So you'll see, and sorry, this PDF document is now also available on the Google Drive. You'll see it's called, I think it's called Reviewer Guidelines. And the intention is here, every single proposal you read is written in a slightly different manner. So as a reviewer, it's quite difficult. So you have to be familiar with what are we looking for, okay? So this is what we look at to have in the back of our mind when we start reading applications. What key information do we need to start pulling reference to, okay? So if we start from the start, the project background. Project background is important because it gives us the context as to why this proposal is being proposed, why it's important, what is the rationale behind what is being proposed. This is where the scientific information comes in. And in the case of the AMI proposal, we scored everything between 0, which was not good, was very bad actually, and then excellent, which was 3. So there was 4 options, 0, 1, 2, and 3. Okay, so the project background also provides an opportunity for the reviewer. In the case of our AMI grants, we were also not expecting entirely fully developed projects. So we needed to understand the full scope of the work so that we could also help convert those proposals into final polished products. Okay, so slightly different to what your task was today. You were tasked with giving a good fleshed out project proposal. We weren't actually expecting that with the first AMI call. We were hoping for it, but we were leaving a little bit of room for project to be developed once it had been accepted, based on the scientific background, okay, the justification and everything like this. All right, so if we look at the project background, it's easiest to look at what we're looking for, so the positives. So if you look in the right-hand column, all relevant information to understand the objective and to support the proposal. So does the project background include enough information for you as a reviewer to understand why and what this project is trying to do? Okay. The next key point is that preliminary data is used to contextualize the proposed experiment. So the proposed experiment doesn't stand in isolation. It refers to something that you've done in the lab, something that's mentioned in the literature, something that has led you to want to do this experiment. Okay, what is that? As an applicant, you need to convey that, and as a reviewer, I have to be able to see what this relevancy is. Okay, and if you contrast that to the negative, it is difficult to determine why microscopy is being pursued in this research. It's a very flat statement, but it happens. People propose things, but there's no context as to why. We spoke about it with Leong. We spoke about it with I4A. Just because an opportunity exists, we see the cases of people apply for the sake of applying and they don't know why. So this is the first hurdle point. The next is the aims and hypotheses. So again, in regards to the visiting researcher program, we were being very selective of the projects that fit good, justifiable biological questions that demanded the microscopy. Okay, we are trying to support quantitative microscopy, and we're not doing things for the sake of doing them. We try and stay away from situations where we're looking and seeing. Sometimes it's good, you need to explore what a sample is, and sometimes you need to just look and see, and then you can develop your hypothesis. But we were trying to support hypothesis-driven research in this case. So the hypothesis and the aim was very important and equally weighted to everything else that was reviewed. So starting again in the positive, in the aim, all the necessary details required to infer an appropriate microscopy experiment and its design. So the aim needs to speak to the experiment that needs to be conducted. If my aim is to cure HIV, can you infer an experiment from that? No. So in the eyes of myself, in the eyes of Ami, that's not a suitable aim, okay? You have to be directive. Your aim has to tell us what you're trying to do, okay? So as a reviewer, all the information was necessary for me to understand as a naive person that's not familiar with that biology, what they're trying to do, just in the aim, right? And if you can convert your research into a clearly succinct aim, you've done a very good job, and that's step number one. Okay, the next is hypothesis. The hypothesis needs to be clear and convey all the relevant information of both the microscopy as well as the microscopy experiment at hand. Again, I'm going to cure, or my hypothesis is that the drug treatment cures HIV. Okay, in theory, that's a hypothesis, but that doesn't, it's nothing actionable. We can't do anything with that. So your hypothesis needs to be more directed, needs to say, in this experiment, we are going to see this result based on this biology. That is the hypothesis. It needs to be firm. You need to be willing to stand on that, right? You have to make a line in the sand where this is what we think is going to happen. Doesn't mean you have to be true, but you have to be willing to say, this is going to be the outcome that we're trying to test for. Because again, in building and developing this experimental pipeline, it's always to the demand of trying to address this hypothesis. So your hypothesis directs how you design your experiment. So it's vitally important that you convey your hypothesis correctly. Okay. On the negative side, there's too little information in the aim to describe the intended goal of the experiment. I conveyed that with my example of curing HIV. Same thing with hypothesis. It's too vague to provide guidance into what experiment needs to be conducted. I believe that XYZ mutation causes increased cell proliferation. Actually, that's not a bad one. That was a terrible example. That was a good example. But you see how I use the word cell proliferation? That's telling us what we're trying to see, and it's increased. That means we have to find a way of measuring it and being able to compare it as being increased or equal or below. just from the aim and hypothesis, you should be able to convey to another scientist what you're trying to do. Okay. All right. Then the next one, again, in the context of the AMI call, I tried to make myself available as much as I could. It was over 250 hours in which I tried to consult with experimental designs, but not everyone did end up consulting with me. But put yourselves in the shoes of an applicant, as a reviewer. On the positive, from the experimental design, you need to see that all the appropriate strategies and tools exist. Okay. Is the sample prep there? Is the way that you're labeling the fluorescent molecule present? Is the drug treatment condition explained? What kind of microscopy are you doing? Is it live cell microscopy? Is it fixed microscopy? What kind of quantification do you need to do? How many samples do you need to look at? These are all questions that a microscopist, that is not inherent to microscopy. It's inherent to the biology that's driving the microscopy. So you as the applicant have to provide this information, but you have to provide it in a way that the reviewer can find it easily and understand it, okay? So that's why in this case, we asked for a clear experimental design. Didn't have to be incredibly detailed, but you say, I'm taking HepG2 cells, I'm culturing them in this media, I'm going to treat them to this condition, this drug at this condition, I'll have a control, we'll fix it, we'll label it with these 4 antibodies, with these 4 fluorophores, and we'll image it on the confocal microscope with the intention of measuring X, Y, Z. Almost on a bullet point basis, yes. It doesn't have to be incredibly specific to say we're going to use 500 micromolar of antibody A and, a 1 in 500 dilution of antibody A, etc. But we have to understand what you're trying to do. And in the case of this proposal, the reason we did it because we were trying to plan a project that would be performed for the first time in a different country, and we had 4 weeks to do it. So the more granularity that we had and clarity we had, the better we could prepare for the project. So again, this is in the context of the AMI proposal. That's what we needed, okay?
So there are quite a few of you who wants to do live cell imaging, right? So all of you hopefully understand that if you're going to do live cell imaging, you cannot rely on antibodies. Your protein of interest has to be tagged with a GFP or any fluorescent protein so that you can watch that cell live. But if you in your proposal, you don't even tell us what fluorescent protein you're going to be tagging your protein of interest with. You're just going to say, we need spinning disk confocal, for example, to follow this cell life. That will result in a rejection because we have no idea what protein you're going to be looking at, what you're going to be following. Do we even have the laser to excite your fluorescent protein? If you don't tell us, we cannot support that project. So that detailism. Does that make sense?
And to add to it, it's also context specific. So in the case of a fluorescent protein, you need a construct to exist. AMI can't help you make a construct. That's a lot of effort. We can't support that. But we can support by providing secondary antibodies. So you don't need to know what secondary antibody you used. You just say, we want to use this primary antibody. So yes, you have to provide specific information in the context of a fluorescent protein because it demands such a specific selection. But an antibody choice, that can be left open-ended, and that can be left open-ended to fit our microscope based on our input. But at the same time, if you say, I want to take one sample and label it with 13 different antibodies, then that's a challenge. Like, it's a little bit too much. So another reason for the experimental design and what we used for the internal reviews, the experimental design helped us work out if this would be feasible in the lab, okay? And remember what Leong said, feasibility is actually a good thing. Don't plan a PhD project to be performed in 3 weeks. That's never going to work. The intention behind the AMI call was to do one experiment. Maybe if you had good enough justification, a related experiment could be included because it was similar sample prep or a similar pipeline that had to be followed. At any point, anyone can stop me and ask a question. Do you want to speak into the microphone?
So When you spoke about aims and hypotheses, there's always this thing in my head where I'm like, do I give an overall umbrella aim and a small aim that makes sense for 4 weeks? Or no, you just stick to the fact that it's a 4-week imaging thing?
So, so that is an excellent question. And I think I want to, before I answer that, I want to address that the AMI call was not a grant. Okay. I know some people get confused and they call it a grant. We're not giving you a grant to perform research. We're giving you an opportunity to do an experiment. So in our context, we care about the experiment. Okay. If you're applying to a Wellcome grant or an NIH grant or something where it's a 5-year project, then they care about the overall umbrella and you have to be very high level. In our case here, we care about one experiment. We want to know what this experiment is about. We don't want to know about the 13 other things that relate to it unless it's directly relevant to the microscopy or justifies the importance. Okay, does that answer the question? Okay, last question.
About experiments, because in several research works, you talk of different levels of approach, from the low to the high. So is the aim of the army is just one experiment, whether it's published or not? Is that because if you want to publish an article, usually maybe you can start from light microscopy. You go to maybe immuno IHC. You want to evaluate the same findings with confocal. The same thing you looked at light. So that's why I'm getting confused.
That is a very good question. So the question is basically, is this only one experiment or what about the entire research endeavor that has to be pursued? In the eyes of this proposal, we were looking to support one experiment that helped contextualize other assays that were performed, whether it was genetic sequencing or microbiological assays or animal studies or something like that. It was meant to be a supplementary experiment that provided insight for the purposes of either supporting a grant proposal. So that was something that we were very clear about, is that if this experiment helped prove a concept that then allowed you to write a grant to pursue this with more money, then we would support it. Or if it was one final question that you needed to complete a research question and publish it, that's what we were looking to do. We do realize, and we have learned, that this wasn't an ideal approach, okay? Doing one experiment in isolation is incredibly challenging, which is why there hasn't been a second call, okay? All right, we learned that very quick. But for the purposes of today's workshop, of your goal and your objective in reviewing it, as well as that you have to review a single project, a single experiment. Okay, it's a good observation, and we're on the same page, actually. I think everyone's on the same page. Okay, back to it. The quantification strategy, also to provide a little bit of background, we were expecting that the quantification was the most challenging to the community, so we weren't expecting a lot of information here. So in the real proposal review form for the reviewers, this first column was not clickable. We did not negatively score anyone that didn't have a clear quantification strategy, but for the purposes of today, we are looking for a quantification strategy that clearly exhibits an understanding of the measurement approach as the best outcome. So what is it that needs to be measured in those microscopy images and how is it going to be measured? Okay, that's what we're looking for. On the negative side, there's no link between what somebody has proposed and what they want to measure. They want to take a sample and put it on a microscope and do super resolution microscopy. Then they don't know what the next step is. We can't support that because there's no justification for what we're trying to measure. What is the rationalization behind that? If you can't explain what you want to measure, then you shouldn't be doing a microscopy experiment. And I understand that there's a nuance between exploratory microscopy, and that is a separate issue that we can talk about at lunch or dinner. But for the purposes of this, if you can't put into words what you want to measure from an image, you shouldn't be taking that image because then you're just taking a pretty picture, and that's not what AMI stands for. Okay. All right. We're halfway there. Actually, a little bit more than halfway. Right. Then we get to the reviewers who are asked to assess the scientific justification. So starting in the positive, there is no other means of gaining the necessary insight into the biology being studied. So this was meant to encapsulate both the justification for the biological engagement, sorry, why this biology was being investigated, but also the justification for doing the microscopy. We were offering to do a microscopy experiment. We needed to make sure that the experiment to be done needed to be a microscopy experiment. Because microscopy is expensive, it's difficult, takes time. And the first thing that we will do when we have a project consultation, I think I chatted to somebody yesterday and they wanted to do a microscopy experiment, and I said, actually, you're better off doing flow cytometry because it's easier. It gives you the information you need and it's much easier. This is what the reviewers are assessing at this point, the scientific justification for doing the microscopy, okay? So I always harp on about this. If you're doing a microscopy-based experiment, you need to support why you're doing that from one of two things. The first is that you need spatial information. You need to measure something in 3D space or 4D space, because it could also be time. You need to measure how far something is, how long something is, how shaped it is, how far it moves, something that exists in 3D space, okay? Or alternatively, you need to measure something that demands that you maintain the biological context. You're not able to dissociate the cells because you lose the feature of interest, okay? If one of those two things are done, you have a firm justification for doing a microscopy experiment. And that's what gets reviewed at this step.
If you score high in all of the aspects except for this one, would it be just cancelled as is? Does the marks then matter?
Yeah, so the scientific justification was one of our non-negotiables. If you scored 0 on this, your project was rejected. Because if you could do it with something else, and you'll see on the nuance side, on the negative, other assays will provide the same information but with higher confidence or reduced cost. So I'm not saying that microscopy has to be the only way to do it, but if microscopy is not the easiest or the cheapest way to, or best way to do it, then it gets cut. Okay. There was another. That's just the online.
Okay, so the same question, but in a different, because I don't know your priority. Let's say we had a consultation session, everything is clear, but the application had like some feedback from you that you think should be changed. Like you accepted the 4-week scientist, but you think it's over ambitious. I need to edit it out. Is that an option?
Yeah, so again, the expectation was that we consulted and we worked with people before they proposed, but a lot of people didn't. So most of those people failed, but we had a reason why they failed. But the people who did consult us, we would go through these things, maybe not in this format, but we would talk about it beforehand. So they would start already trying to manage the project to be aligned for a specific reason.
Okay, so even regarding after the consultation and you get all the feedback from the team or whoever you're applying, the AMI or the AIC, you can even send the proposal. We can review it and then we give you the feedback with all of the feedback you just adjusted and then resend it again. So it's not a grant application, and Mike were saying, we're working so hard for people to be accepted and to do the experiment that they want to do, if they are collaborative.
Yeah. In the case of AMI, I think there were only about 9 people that actually submitted the proposal beforehand. And 3 of them, it was actually 4, they sent me 4 drafts. And I revised it, commented on it, and sent it back. So again, the AIC does the same thing, AMI does it. Doesn't necessarily mean it's applicable to a grant, but You use what is available to you. And in the cases of these two situations, we were available. Okay. All right. The next one, which was the really driving factor for AMI, was the personal motivation. Why should you, as this scientist, coming from an African institution, why should you be selected? Okay. And this came down because we were providing these tools, but they were meant to be for the whole of Africa. So we wanted there to be this criteria that prevented it being overcapitalized on by researchers that came from high resource environments to actually allow research to proliferate into the continent. So in the positive, there's not much of a positive because it could be any justification that you can motivate. But in the negative, similar imaging technologies are available at their home institution or collaborators' institution. So if you were a prolific researcher and you had lots of collaborations in the US and in Norway and Australia and Japan and all sorts of things, and you were proposing to do wide field microscopy, you could have the best proposal ever, we would say no. Because go work with your existing things. You are already well positioned to take advantage of microscopy. We were trying to support those that weren't. Okay. So sometimes there's a personal motivation. And in AMI and in the project, the proposal guidelines, you had to indicate what other microscopy tools do you have available to you so that we could understand, oh, you're coming from the University of Cape Town. We're not going to let you use it because you could use the core facility. Okay. All right. And then the last thing is the direct impact. And this is the impact and the ultimate justification for investing into the project. What would result from this? And this was speaking to what Kashmir brought up earlier. In the positive, novel the experiment and the line of inquiry is novel or has never been applied to the research question at hand. So that means it's either never been done, or you're taking something that has been done in a setting that's not Africa, in a research question that's not relevant to Africa. So let's say cancer biology. I'm not saying it's not relevant, but there's a lot of cancer biology elsewhere in the world. And you want to apply a certain line of investigation, but this time apply it to tuberculosis. That is novel, okay? That's the kind of thing that elevates the scientific question and helps push that meaningful outcomes come from the time at AMI, okay? And then it goes on to highlight that the results could have considerable ramifications on either the lab or the applicant. So it could mean that it helps justify a grant proposal. It's a preliminary data. So we don't necessarily only support things that would be published. It would be somebody who says, if I can validate this thing works, I'm better positioned to apply for a grant. Or it could be for a publication that I have a 90% complete story. There's just one uncertainty that we have, and the reviewers has potentially asked us to address this question. That is the direct impact. That this work at AMI will have a direct result in allowing me to get to a publication, allowing me to get to a grant proposal, something like that. Okay? The negative being the impact of the experiment is marginal. This happens. People apply just for the sake of applying. They're doing things just because they want to learn. AMI supports learning, but not through the visiting researcher program. If you want to learn, attend a workshop, apply for a shadowing, etc., etc. This money was allocated to support research, so we wanted to ensure that. And the last point being, the project will likely only be confirmatory of other experiments or assays performed, which means that the focus is not on the microscopy. Microscopy is not actually bringing that much to the table. You've already got 90% of the way there. Either you should just keep repeating until you get statistical significance or something like that. But microscopy is not bringing anything new to the table. Okay, so that's the last point, the direct impact. Any questions about any of this? Because you're going to start reviewing each other's proposals based on this now.
matter for this round, but it's good for us to know because Leong told us about the availability of reagents in AIC, and I just want to know, like, when you say there are secondary antibodies available at AMI, is there a way of knowing what is available a priori?
Not unless you have a consultation with
Yeah.
us.
Okay.
Yeah.
So actually, everything I've heard so far is like, we're gonna hammer you with emails, both of you.
Pretty much, yeah.
Yeah.
But if an email contains, "Hi, I study this thing, what can I do?"
The last point made me think of a scenario where you've got a lot of the information, but then your paper publication is not accepted until you do a microscopy experiment. Does that get accepted if it's well enough written?
If you're motivated, yes. A reviewer has asked specifically for microscopy or asked for a situation that we believe microscopy will help address. Perfect.
Okay, thanks.
Sorry to those online. Is it still low?
So what I was saying is that this is an opportunity for everybody to gather up your courage and ask questions, because we're actually really hoping that you guys can ask us as many questions as possible. Because in real life, as I've mentioned earlier, you actually have to pick up the phone and call somebody that you don't know to ask questions. So this is a great time for those who have not asked the questions to ask the questions. Trust me, there is no such thing as a stupid questions. If you ask, people in this room as well as online will be extremely appreciative that you actually ask that question. At the back. I'll let
So I'm still a bit confused about how to approach an imaging technique. So my knowledge, for example, goes up to fluorescent microscopy, even though I know that what I'm investigating in the lab, I can visualize in a better way, high resolution, but that's where I end. It's like, I know it should be a high resolution technique because I need to see some detailed structure of some elements in the cell. But then I always like hesitant, it's like send you an email. I'm into, which you just mentioned that that's not like, don't send me something like that. I'm interested in this organelle or protein or whatever. What should I do about it? So it's like how to, and I open so many articles. It's like, would this give me what I want? And there is a level of information I cannot really comprehend beyond
yes.
the basics of my knowledge. So where to start when you're really confused about which technique to use, and then you go from there into designing an experiment.
Thank you for that questions. That actually got to the gist of what we're dealing. Is we actually don't expect any of you to actually have intimate knowledge of microscopy. And this is what I'm going to repeat. I know I'm sounding like a broken record, is to talk to us, talk to us, talk to us. And one of the key discussion that we have just outside is somebody is trying to shoot for resolution microscopy because they cannot see some sort of an aggregate. And we have a very important discussion whether or not this is a detection problem or this is a resolution problem. And do you always have to chase after the highest resolution possible? And the answer is no. In fact, if you are trying to see if protein A and protein B interact with one another, going for the highest possible resolution is actually counterproductive. Now, you will never know that because you are not trained as a microscopist and you are just trying to approach us to use a microscope. That is why you talk to us. And the best way to approach us is truly just, or even imaging for all. I know that they are not represented right now. Same idea is I'm very interested in pursuing these questions, and I know that only microscopy can help, but I'm not sure which instrument or which technique I should use. Can you provide us some advice? That's all you need. That is how low of an entry energy that you need to overcome is tell us your biology because you are the expert in your biology. Tell us the biology and then ask us for advice of which instrument to use or which technique to approach. All of us will respond within 24, 48 hours. And that is how important that is.
Even if the facility is not going to support your hypothesis or your question with the microscopes that you have, we're going to advise you which facility do you go in order to get the microscope to answer your hypothesis question. So,
and because I know that I said something that maybe I didn't explain enough. What we don't want to hear is somebody saying, I work with Plasmodium, what can I do? Right? That's what we don't want to hear. But if you give us more information, you say, I'm looking at this specific thing in this environment, we think this, what can we do? Then that's much easier to tackle. And I also think that there's a level of ownership that we have to take on as a community to not be 100% naive to microscopy. See what is done in the literature. What have other people done in similar research fields? You know, you can learn from the cancer field and apply it to the autoimmune field or etc. You don't have to be in isolation, right? And don't be afraid to say, I saw a paper, it looked quite cool. Could we do something like this? That's also a great thing for us as microscopists to work with. Karen.
Can I give another example to try and understand this? So, slightly independent of AMI, but it was a query that came via AMI, was someone to me wanting access to microscopes that we have in Cape Town aside from AMI. And the email was something along the lines of, hi, I'm interested in collagen in quails. Can we chat about what microscopes you have? I'll be honest, I have absolutely no idea how to respond to an email like that. I ended up having a call with him, and it was something like they were interested in the formation and organization of type 4 collagen fibers in the growth of certain bones in quails under certain conditions, and they had access to certain types of samples, and they wanted to know if there was a better way of visualizing or trying to do something that they had tried before. It makes my life, Leong's life, Mike's life really hard if I get a, hi, I'm interested in collagen in quails email. But an email that said, hi there, I'm interested in type 4 collagen fiber formation in quails. I have access to embryo bone tissue sections. I've tried X, but it's not quite giving me what I want. Or maybe you haven't tried. I'm wondering if I could try. Could we set up a chat? That immediately Mike or I or Leong know how to respond to that. We know what we're going into when we go into the meeting. We know if we can help you, or if we know that we can't help, but I think someone else can. So you don't have to know about the technology, but try not to make our lives hard when we're trying to help. And another point that I want to pick up on quickly, Mike used the word measure in terms of quantification. And a little bit of a second story from me. When I was a PhD student and relatively naive, I visited collaborators to use a more advanced microscope than we had. And they kept on referring to my time on the microscope as taking measurements. I was like, I'm collecting images. They're like, no, you're taking measurements. No, I'm generating images. Measurements. It's a weird cognitive shift. That if you go, oh yes, I am measuring, whatever you're labeling, you can see it, you can see where it is, you can see if it moves, you can see where it is in relation to something else. If that helps you think about what you're measuring, put that into your question when you're contacting and asking someone for help or writing a proposal. You're not collecting images at a microscope. You are collecting incredibly rich quantitative information in 3D space.
Okay.
That's
So,
my 2 cents.
yes. Yes, new face. Awesome.
Okay, I just would like to ask if I have an experiment with a timeline like 1 month. I need to image my cells after exposing to a specific drug for a 1 month, and the total period of the grant is only 2 weeks. So can I extend this or not?
So that all so basically the idea, let me generalize it a little bit. So let's say if your sample preparation to set up that experiment takes a very long time, longer than what the imaging center allows you. There are several ways. Either, so it all depends on the sample and depends on the country and depends on the imaging center. So I'm just gonna generalize the ideas. Some imaging center actually allows you to arrive way early and just not doing imaging, just preparing your sample. AIC is one of such imaging center. Some people go there to build organoid, and you may need to build organoid for 2 or 3 weeks, and before you can image. So we can support people who go there, not the whole team, but one person go there to start building organoid on a daily basis, so that downstream every day there is a fresh organoid to be used. There are also imaging center, especially if you're just dealing with cells, is to send the cells there early. We tell the imaging center exactly how you need to treat the cells, and we can treat them in advance for you, especially if it's just a monolayer of cells. So there are always various ways. Or if you're doing fixed sample, you can treat all of them at home, fix all of them, and just ship the slides, right? So there are a lot of variety to do that. Absolutely.
So if I have to extend a specific experiment based on a reviewer comment or something like that, to that I have a timeline, and for the time being, the grant is not open for application. So can I mention this, or we are just restricted to the timeline for the grant?
That also depends on the program. So some program has very strict guideline. I'm not sure if AMI has that. AIC does not. Even though we have specific open call for proposal, we actually reserve some scope time for ad hoc applicant. We all know that at some point review is going to ask for something crazy. You let us know, we will bring you back in very, very quickly. And it all depends on the program.
Yeah, the same thing applies to AMI. We can't promise anything, but if you get in touch with us, we can see if we can help, given the timeline. On that note, we have to call the session because we need to go to lunch, because it's also poster sessions. Leong wants to say something.
So I want to sort of give you guys a morning observation. After my talk or during my talk, almost every slide, I basically say, please talk to us, please talk to us. You guys are sick and tired of hearing it. I'm sick and tired of speaking it. So in the morning session, all of us were walking around the table. Very few table actually raise your hand and say, hey, we have this idea, we have this proposal. Do you think this is acceptable? What else can we change? Not a single table took advantage of that opportunity. That makes me sad. I don't know what more we can do to encourage you guys to really, really, really talk to us. Please talk to us. Take advantage of it, right? And we talk about, well, maybe it's a cultural issues. We have been rejected all the time. We never really get a chance to talk to people. We're walking around. Sometimes I was sitting there staring at you. Nobody talked to us. Change that attitude. Seriously, change that attitude and talk to us. I'm here. We're all here to help you. At the end, finally, the last few minutes, people start saying, is this okay or is that not okay? Be more aggressive, because I can guarantee you, you don't talk to us, people in Europe talk to us. People in the United States talk to us, and they are in a much more privileged situation. They're going to beat the hell out of you in that proposal process. If you want to win, you fight. You fight. You do not deal with this passively. You fight, because you are fighting against the whole world. Fight. It is yours to take it.
Welcome back, everyone, and to those online, welcome back and thank you for your patience. We're going to enter the review process. So, Leong, are you leading this?
All right. So everybody has access to all the proposal, right? So when the two designated group come down and discuss their opinion, one more time. All right. So when people come down and review their proposal, feel free to open up that particular grant that is being reviewed so that you can ask questions. You can very quickly scan through it and then ask questions. Now, there is a tendency for those whose proposal is being reviewed to raise their hand and defend their proposal. You don't have that luxury in real life. You just have to shut up and take it. But bear in mind that we're all here to learn. Please don't feel embarrassed. This is a learning process. I myself have gone through this learning process. So this is a good way for everybody to learn. In fact, your mistakes, if any, are extremely valuable for this process. So please don't feel embarrassed, okay? And then we'll discuss it, we'll dissect it, but please don't feel that it is a personal attack or anything. We're just all here to learn. All right. Who review group one? Come on down. People who review group one. We don't have a lot of time, so please come down. Please come down. Who else review proposal one? Proposal one. Either the whole group come down or the designated spokesperson come down. Come on down. Come on down. Okay. And as I said before, this has to be a very constructive process. Please don't be rude. We are all scientists. We are all trying to help one another. Even if the proposal is not good and can use improvement, you focus on the can use improvement part and not the shortcoming of the grant. So whoever wants to go first will be the first reviewer. Shoot.
All right. So group 6 here, excuse me if I'm being too generous or too strict, first time reviewer. So the proposal that we reviewed was talking about the effect of plant X on sperm count or fertility in certain regions in Asia. And they were proposing to test plant X on mice and then assess the morphology of the spermatozoa and the functional effect of the spermatozoa using a target called arcosine using confocal microscopy. So for the project background, we gave it a 1. And we felt that the summary provided enough context for the experiment to be done. However, there was little information on the background experiments that were mentioned. There was just mention of using light microscopy and genomic techniques, which we felt lacked the depth to understand what was the context for the previous experiments to justify this one. However, all the context regarding the why the experiment was done was there. So we felt 1 was a fair assessment. And also they did provide context for why they wanted to quantify arcosine, which is the target they wanted to probe to test functionality. Yeah. And regarding the hypothesis and aims, we felt 2 was a fair score. The hypothesis was very well formulated, it was specific, and the aims were very clear. But the description of the results that were expected were lacking, especially the arcosine, which is the target that they wanted to test for the functionality of the spermatozoa. It was mentioned in the background, but they didn't mention what they expected to see as a result in the hypothesis. And for the next section, the experimental outline, as someone who is not specialized in the field, some terminology was mentioned that I wasn't sure what it represented. So they mentioned that they wanted to collect the epididymis from the mice, and I had no clue what that meant. They also mentioned the time frame of the experiment where the mice will be treated for 3 months and then do the immunofluorescence experiment. So as someone who would be giving the grant, as though I'm very rich to provide that, it just didn't feel practical in terms of timeline.
Are they going to do those experiment at home and then fix the tissues and send it over?
Well, there was no specific details, so it just felt like the lack of specificity was like we deduced points for that. Also, to generate the positive control, they mentioned that it will be done through a sonication process to introduce the primary and secondary antibodies. Also, as someone who's not specialized in the field, it just didn't make sense or I didn't understand. So a little bit of background would have been helpful. But they did include like a reference paper to describe the process. So maybe with
That will consider, if it is published, we'll consider that acceptable.
yeah. Well, yeah, I didn't look at the reference, but it was referenced, so I think this is a
Fine.
point for their side.
We can give them point for that.
Yeah. Also, they mentioned that they wanted to test 30 slides in the microscope, but they mentioned that we need at least a number. They mentioned something along the lines that we need for quantification at least 30 sperm. So does that mean 30 sperm cells or what exactly? I feel like the term 30 sperm was a bit general. Like I don't know what's the unit here. So I think it maybe needed a bit more specificity. The rest of the sections, we got caught up in a lot of discussion, so we weren't able to grade forward. But as an initial impression, we felt that there was a strong background being provided, but with some small details, like it would have been a stronger application. So please try to talk to us and get more information.
Outstanding review. Thank you. Thank you, reviewer one. And usually you don't get the applause at the review panel. Reviewer two.
Yeah, we agree with most of what was said from group 6. Okay. But regarding the positive points, we found that the personal statement was good. The problem in general was relevant, and they put attention to detail in describing the experiment itself. What was the hypothesis was a bit too generic, and we did not really understand why the high resolution microscopy was needed. And we also did not understand why the group chose tubulin and acrosine in specific. Do you want to add something? WM.
Anything else?
That's it.
Okay. Okay. Now we're opening the floor for discussion and questions. Other than group one, anybody can open their mouth. Because group one, you are not in the room, supposedly. Any questions? Do you guys agree, disagree? Sounds good? I think that's fine if nobody has any. By the way, both reviewers did a wonderful job. So thank you. Not bad for the first time, actually.
So
Are you guys discussing funding? What the hell?
sorry, I was just checking that I hadn't missed a comment being made. So I have a question. I obviously didn't read the proposal, but if I understood correctly, that this was a proposal to study a plant, the impacts of a plant on spermatozoa formation or function from Asia. Given that this is a proposal for the Africa Microscopy Initiative, does it fit the remit of the purpose of the initiative, or was there sufficient information in the impact section of this work as to why this should be done by Africans in an African facility?
I have an answer for that, but I want to see if you guys can address that. Everybody understand the questions? This is a proposal to study the MSG effect on spermatozoa, usually in the Asia community. And Karen's question is, why are we using African resources to study something that is more relevant to the Asia continent?
They mentioned briefly in the last section, which I vaguely remember now, but they included a statement saying that this is one of the only labs in the region that study fertility tests and the function of the spermatozoa. So maybe by studying this, it could give some insights into how this could be applied for a similar issue or a similar problem within the region. Maybe this is Asia specific, but at least in terms of technique and how they study it, it could have implications for something that they would do in Africa.
Anything else you want to add?
I think the problem itself is relevant, but it was not really well shown why high resolution microscopy is relevant here in
Okay,
this
so
case.
two questions there. So if I may address Karen's question. So Karen set up a very good trap here, is any imaging center is not there to question the national funding direction. If this project is funded by the country, whatever country that may be, we're not here as an imaging center to question why this particular country is funding something that is related only to Asia. That is not the point of the imaging center. We are here to really empower African scientists to do their science, right? So I think that is fine. If you're not sure, especially if you have this kind of conflict, you can always say there's this grant or whatever is supported by this. Then that basically eases the problem right away, right? So we usually don't question that. Give me 1 second. I'll ask. So reviewer 2 raised a very important point. If you read that proposal very carefully, the justification for confocal microscopy is not very well written. It says that confocal can give you better resolution. The reason why you want to use confocal now, the use of confocal is not wrong, because they are using tissue samples, and tissue sample used to be thicker, so there's a lot of out of focus light. Therefore, you use a confocal to reject out of focus light. Confocal is not there to give you higher resolution. It will give you better contrast. Therefore, you see images better. So, but it is also important that for whoever is writing this grant to say that we have used bright field microscopy, and right now we see a lot of out of focus image. We need confocal to deblur that image. And now in reality, when you have like a week or two or a month to write that proposal, you will include preliminary data image and show that your bright field or white field image is just not sufficient to address the question.
I do want to flag we're 15 minutes in and we're 1:12.
That's fine. Keep going, keep going. It'll go faster. Okay, you guys can go back.
Yeah, I will try to go fast. By the way, we are group 1 here. So first, so first we mentioned that the lab did some biochemical assays, but it wasn't enough to answer our question. Although they tried light microscopy to see not the tissue, we are just seeing the sperm. It wasn't super critical to judge the whole structure, to see the head and the tail, as we mentioned. So they have to shift to confocal microscopy. I think we mentioned this clear in the proposal. Do you think so?
So you have to write that in the justification. I read through the justification.
Yeah, we write this through the justification. Also, we mentioned in the background.
Put it in the right section. Okay, next. I think we're done. You want to ask a question?
Yeah. Yeah, so regarding your comment on the relevance of this project to the African continent, if it was well formulated that this could have stronger implications, would this still be counted as a strong application, or?
Yeah, especially already funded by another government. It's not our place.
Yep, okay.
All right, thank you very much.
And well done to group 1.
All right, whoever review group 2, come down, and we have to do this very quickly. We have to do this very quickly, very quickly, very quickly. Only reviewer 1 showed up. Where's the other group? Okay, then it will be just you. I'll help you out.
Stand there, it will be easier with the book.
This is working, right? So with the disclaimer that we weren't able to finish it with this was the second one we did, so we didn't have as much time. Group 2's application aimed at looking at ALS, a disease called ALS, and the impact of certain mutations in a protein called SOD1 in the Egyptian population and the effect of those mutants on the localization of the protein in the cells and how the localization turns into aggregates, and then how these aggregates affect the mobility of the protein inside the cell. Along those lines. Our very first comment was that the first section of the summary, while well explained, did not have a lot of layperson language. None of us studied this before, so there were a lot of terms that we did not understand. And I think in the format it said that this section needed to be communicable as possible. For the aims and the hypothesis, and going into the design, the plan was to have fixed samples to look at the different mutations that are tagged and then look at their localization. And then also do live cell imaging and look at the formation of aggregates. And also do FRAP to look at the mobility of the protein inside the cell, which we think for the 4 weeks is rather ambitious. In the case of the design, I think the steps could have been more detailed in terms of what they wanted to do, the specific steps. When it came to the actual steps, the FRAP disappeared from the application, so maybe it was there in an earlier version and then wasn't updated in the earlier part. And for the personal motivation, which I think was the last part we reached, it was quite a good a justification that this kind of study needs a confocal microscope, which they don't have, so they want results in order to apply for a grant to get a confocal microscope, which I thought seemed fair. You guys wanna... Did no one else? Sorry. Thank you.
Good job. All right. Review. Whoever review group 3. Oh, that's also you. Who is the other group? Come on down. Come on down. You can start speaking.
Okay. So for group 3, the summary of the application was to look at the relationship between infection with a parasite called Leishmania and the state of the acidification of vacuoles inside the macrophages, and how the progression of the infection happens in different states of the acidification of the macrophage. Overall, we thought it was really well written, but the first problem that came up to us is that the organization of the application did not fit the outline that was given by the funder. So we spent a lot of time looking at different sections and going back and forth between the guidelines and the written to try to find the different parts. So that took time. For example, in terms of the background, there was a section for future applications, which then had another part down for the impact, things like that. For the hypothesis statement, it was well explained, but in 2 parts that could have been streamlined into one. So the hypothesis mentioned that an increase in acidification is likely to cause less parasite infection. And then the vice versa statement was then mentioned. And it could have been said something like, the acidification affects the level of parasite infection, and that's it. For the design, this is something maybe where it's something that we don't know, but part of the plan was that there would be transfer of genetically engineered Leishmania, which is biosafety level 2, And I don't know how feasible that is to transfer between countries. Yeah. And in terms of the detailed steps, the quantification was really well explained, all the details, lots of minutiae, but for the design, the steps, the experimental steps itself were not as explained, which I think would have been more, would have also been required for the design. One part that was not mentioned in the quantification is how the lysosomal acidification or the lysosomal acidification was not mentioned in the table for the quantifiable outcomes, where it is a crucial part of the hypothesis. And finally, the section about the future impact about the metabolic HIF alpha, I think that wasn't really well explained. And from what we heard in the talk this morning, that maybe for this kind of application, it doesn't really feature what the future plans would be. They only want to know about one experiment. Otherwise, it was really well written, and we're very jealous that you were able to write all of this in 2 hours.
Thank you very much, reviewer 1. Reviewer 2. Can you pass?
Thank you very much for review of group 3. In addition of all of those that they see, we have specifically notice that the group, they have a jump from interaction, direct interaction between macrophages and leishmaniasis without going gradually from what is already known microscopically before talking about why they want to use that microscopic techniques. I think that is one important point that we can add on what they have already said.
Anything else? So I think one of the biggest problem with this particular grant is both groups of reviewers got confused because the format does not follow the what is prescribed in the call in the program. You do not want to confuse the reviewer ever because they actually didn't know how to score. And this actually happens in real life. When you change the format, you confuse the reviewer, and even people like me who review it all the time, we actually don't know how to score because we're supposed to follow the instruction. And now if the applicants change the format, then it makes it extremely difficult. Just a quick thing. So I don't think the hypothesis statement is wrong. It is scientifically accurate, but I think it can be streamlined, specifically this part. Basically say during macrophage infection, vacuoles that maintain stronger acidification and higher lysosomal marker enrichment will restrict parasite multiplication, whereas vacuoles with weak and delayed acidification will permit faster replication. Now, this is actually an expected outcome. This is not the hypothesis. A better hypothetical statement is the degree of acidification of the vacuole can affect this and this. And then this is an outcome statement, okay? So this is more of a formatting issues that kind of unfortunately jeopardize this particular proposal. Any other point? Thank you very much, reviewers. Who reviewed group 4? Who is reviewer 2? Come on down, come on down, come on down. You can come down, and if there's anything to add, you can add. You haven't read it at all. Okay, so then you guys can carry the show, and then we'll go from there. Go ahead.
Thank you for group 4 application, talking about reproduction spermatozoids. The title I don't have. I did not copy it.
Wait, what happened?
Yes.
How come there is no group 4 on my?
It's on spermatozoid, male reproductive system.
Oh, the Plan X thing.
Yes, male reproductive system, spermatozoid.
That's one,
yes.
right?
Project 4. Project 4.
Yes.
Yes. Identification of change.
Vimentin as a marker of
Yes.
oxidative stress.
Yes.
Okay.
The first thing that we notice in this project is that the reason for using confocal microscopy was not overly underlined. They just jump and say that they want to use microscopy, confocal microscopy, without explaining why they want to use that microscopy and what is the limitation. Did they start with a bright field microscopy and they have the limitation? They did not explain. The second thing that we notice is the time frame. The group explained that they will be using treating animal for 8 weeks, and after the 8 weeks, they will collect the organ. After collection of organ, now they will do histology. But they did not mention at which level they are going to AMI center. It means that it can be confusing to the reviewer whether they will go do the complete experiment in AMI center or if they are doing the experiment in their lab and just carrying the sample for histology at AMI center. Another weak point of the project is that they talk about oxidative stress, but they never measure oxidative stress, explain how they will have to measure oxidative stress during their experiment.
That's a very good catch, by the way.
So we think that
So let me
even the personal justification and direct impact, we saw that they were weak. The impact was weak because you cannot just say that you want to use it and come back and train people. Oh, yes, you use it, you are trained, but you come back to train people without equipment in your laboratory, so you cannot train people. So we think that it was weak. They could have explained it otherwise. And also for personal is okay. I think that is everything that we have noticed concerning the project 4. Thank you.
Thank you. Any questions? All right, thank you. Very good job. Very, very good job. All right, who reviewed proposal number 5? There is a question online. Can you read it out? Whoever has a mic.
Lillian, you can unmute yourself.
Hello, can you hear me? Hello? Hello? Can everyone hear me?
Yes, we can hear you.
Yes, we can. We can hear you, Lillian.
Hello? Hello?
Lillian, you can go ahead and talk.
Hello? Okay, can you hear me? Okay, my question is this. This particular paper, when I saw the heading, like using bimethine as a marker of oxidative stress, personally, what I know about bimethine is one of the cytoskeletal elements you can find in a spermatozoa structure. So I don't know how the person can show us that bimethine is a marker for oxidative stress, or he wants to tell us a part of maybe antibody that will show the structure of bimethine in the spermatozoa, because bimethine is a part of a cytoskeletal structure found in a spermatozoa.
Thank you, Lillian. Yes, that I think this just speaks to your no, we're going through the wrong mic. So Lillian, thank you for that. I think this speaks to the fact that this was a project that was developed in a short term. So I think we can give it a few benefit of the doubt. But what you raised is certainly a very valid point, that there needs to be a justification for what you're trying to measure or quantify actually corresponds to the biological process that you're investigating. Want to add to that?
Thank you.
this is movement of cells. You know, you have what you call spermatogenic series. The cells keep dividing from spermatogonia, spermatocytes, primary, secondary, and tertiary to the mature sperm cell. So the skeleton ensures movement. The skeleton, the vimentin ensures movement of during cell division into various stages. So there are 3 cells you identify vimentin: the Sertoli cells, the Leydig cells, then the peritubular cells. And all these cells, Leydig cells plays a role in testosterone production. So if they are not dividing with the skeletal components, then there is a problem. There is a problem.
I don't think the cytoskeleton part is the problem. The problem is that you guys mentioned oxidative stress, but we did not measure it. And I think you either remove the word oxidative stress, pose an experiment, then. All right, go ahead.
Can you please open page 3 of the grant?
I guess I can, if I can find it. Yeah, I know. That's the
Group 5.
Wait,
Yeah.
group 5 or group 3?
5, please. 5, page 3. Thank you. Yeah, up in the scheme, please, because it's easier to describe their project like that. Thank you. Okay, so the project title is Live Cell Monitoring of Immune Cell Infiltration and Cytotoxicity in Organoid Culture. In a nutshell, the project is very nicely rationalized. I think it's very attractive. I read it also because of my work in immunology. It was very attractive. It reads very structured. And I was tempted to think it's perfect, right? And I think the devil lies in the details. And the details for me are as follows. This is a 4-week proposal. And it's an overambitious grant in general. Like you want to do 3 aims over 4 weeks and everything is interlinked. If aim number 1 doesn't work, aim number 2 cannot be tested. If aim number 2 doesn't work, aim number 3 doesn't get tested. And I would rather focus on one aim only and prove that it's working. Aim number 1 is based on, the whole project is based on making organoids, which I assumed has been done before. 5 days organoid culture, fantastic if this was done before. But transfection is really the Achilles heel of the project, because where are you going to transfect? Are you going to transfect in the facility? What if the efficiency doesn't work? How about the variability? Like everything is based on comparative analysis, but then the initial starting point is full of variability. So that's a project that would have, at least if I was the funding person, be very happy to onboard if it was pre-discussed. Because it like really the simpler the better. It's just too much for 4 weeks, but the question is very nice and it's very tackleable. And if you edited out all the details, then it would have easily been done. I think that is the strongest point is the project background. The weakest point is the quantification, because you keep on saying detecting, but how? And then the small differences between infiltration and interaction, like are you measuring radiuses? Are you measuring numbers? What exactly is the quantitative parameter that is coming out of the project was not clear to me. Otherwise, it's very nice question and it's very rationalizable. Why would you approach an imaging facility with confocality to answer it?
Very well described. Okay, reviewer 2.
I'm not very familiar with this type of work. However, I would say the project background was concise, so it was easy to follow. I would agree on the, just from the diagram, that everything seemed to be dependent on each other. So again, which made me feel like, just from the diagram, that you're actually doing one experiment, but you kind of split it into 3 different aims, but it's exactly the same experiment. And also the thing that you put the aim and then following the hypothesis, it should be the other way around. That's just a technical issue or in the writing. I wasn't sure, again, maybe that's part of the science, not the, number one, the microscopy, you mentioned that you're using a specific microscopy. It wasn't clear to me as, like, I don't understand all type of microscopy, why you specifically mentioned that. So if there would be an explanation of the choice of microscopy. And also the quantification wasn't really clear to me, like what exactly are we measuring and how, or the analysis tool. It's just taking the image and what analysis tool, it wasn't very clear. That's.
Forgot something very important. You guys said that you're going to use freshly isolated immune cells. As in, are you going to donate your own blood in the facility and use the blood for the experiment to do the co-cultures? Do you have consent forms? I guess if I'm a facility, I would be like, what? Like, I don't know if that works or not.
I think for yes, that's all very important point, but I think we can sort of, give them a little bit of a rope there. But absolutely, I think you guys, both of you, identify probably one of the most, Emmanuel, one of the most, I would say, a kiss of death in any reviewer's comment is the aims are interdependent. That means that if aim 2 relies on aim 1, aim 3 relies on aim 2, most reviewers don't like that, because if the aim 1 fail, you have no project. So try your best to avoid that. So that is always a key thing. All right, any other point? All right, yes. Guys, I cannot tell you how important and how easy to read it is. I know you guys don't have time today, but when you are writing a grant, use a diagram as much as possible, because you have no idea how many words this saved. Imagine translate that into words. Just imagine translate that into words. How difficult it is for the reviewer to even understand what you're trying to do. Just the schematic representation is extremely powerful. So kudos to the team who actually spend the time to make the graphics. Awesome. Okay. All right, group 6. Wow, we're down to the home stretch.
For this project, well, quite a lot to read and quite a lot to understand. A lot of background, and I think they dive into yeast work, which is not a lot of people actually do yeast work. So there is a lot of background. I don't know if all this background was important to go into the specific aim. Again, and there is a jump between using a yeast, and then when you go to the hypothesis and specific aim, you realize that they're using a mammalian system. So it wasn't a very clear why they did this, even though I see the justification, but just because of the amount of text that you have to go through, it was just a bit overwhelming. And then you have the hypothesis and the specific. So the hypothesis is well written, but how they answer, it's quite a lot of experiments, which seem to me this is part of a grant. And this is not a project that was written specifically to that. And they're trying to tweak the grant or a big aim in a grant and put it into that proposal. Because apart from having some of the channels that they may not really work, the actual work, it include live cell imaging, multifocal, I think they use FRAP. So there's just quite a lot. And again, you go through a lot of text and you kind of lose the focus point. What are we trying to achieve for this particular visit or for this particular aim? So even though it's kind of seems like, oh, this is nice work, for this particular application, you get lost. What are we doing for AMI or for the application? So this is in a nutshell how we, even though there is validity in a lot of the things are written, but It's just quite a lot.
Got lost in the weeds.
Yes, yes.
Just I wanna thank for a lot of information from the project, but actually we have a problem with the background. We see that just jumping from one information to other without a clear specification. So the aim of the project is that they need to test the localization of a newly discovered protein and the role on DNA repair. So you just to start with mention what's this type of DNA repair and what is this gene specifically and the role of this genes. We were completely lost. So we give it to for the hypothesis, I disagree with you. We thought it's super generic. If you, yeah, just you can say in the hypothesis we want to test the role of baladin on this type of DNA damage, or you can say we hypothesize that the baladin has a role on 2-BCC repair, and that's it. So this is very long. We give it, sorry, one. For the experiment design, there is just a technical comment. For the use, can we just go down for the experiment? Yeah, the usage here of YFB and GFB, they already overlap with each other. So you will have a post-positive, a false positive. You will not be able to determine if the co-localization because of the overlap between the signals or because the two proteins already co-localize with each other. So you should ask first about the combination of the dyes, if this combination is doable to be used in this experiment or not. So we give it 2. What else? Yeah, for just this justification, yeah, the context is not super clear and not consistent. We need a consistency. There is a problem in the whole grant that there is not a consistency in writing. So you need to test the co-localization. So try to justify each item you want to test. We got completely lost.
I think the consistency part
Yeah.
can be more forgiving because everyone is dividing and conquering the whole day. So you're going to have multiple voices in one proposal, which is unique only to this exercise.
Okay, for the impact, they only mentioned the impact on the lab. They didn't mention the scientific impact or the global impact or the impact in the country. So just add this part. And for the impact on the lab, yeah, it was very clear, but just add much information. I think we have something else.
All right, we have to wrap it up.
Do we?
Wait, I have to wrap up.
Yeah, for the quantification section, yeah, there is a lot of parameter to be tested. Just try to be more specific. You want to measure the intensity and the localization. So if you want to measure the intensity, you will measure the more recruitments of the protein. But you are here testing the localization, if this protein localized to the DNA damage or not. So try to be more specific. Yeah, and that's it, sorry. But actually, yeah, if you just separate or take the most important information from the grant, it would be fantastic. Just try to delete some sections, it would be fine.
Okay, thank you. Thank you, everyone. Couple of things, which is actually very important. Thank you. I'll hand this to Mike just in case anybody want to ask questions. Couple of things, very important point. So you hear a lot of the reviewers come out and say, I'm not the expert in this field. I don't know enough about this field. In reality, people at the Advanced Imaging Center, at the Army Imaging Center do not have the luxury of saying that I don't know this field. We absolutely have to know the field. Otherwise, it is extremely unfair to you as an applicant. So you can make our life easier by writing it in a language that people can understand, right? If you're not sure, hand it to your friend or your colleague who is not in your field and see if they can understand it. And read out that what you have written, read it out loud and see if it makes sense to you. And ask yourself, every sentence, can it be shortened? Can it be made any simpler? Do I really have to use big words? Can I use smaller words? That's number one. So we don't have the luxury of saying, I don't know this field. We just absolutely have to review it. And so if we don't know, we will spend a lot of hours researching just until we get it. So that takes up a lot of time. The second thing, I want to congratulate everyone. I just spoke with Mike. Even though you guys are given only 3 or 4 hours to write, this is exponentially better than any of the proposal that Army Imaging Center has received. Awesome. Oh, you should give yourself a big hand. Congratulations. Now, I hope this exercise is helpful. Okay, good. So now let me give you a couple of tricks before we wrap it up. Now, how many of you have watched Star Wars before? The movie Star Wars. Most of you have watched Star Wars before. So you know that in Star Wars, there's this Jedi that can control people's mind. You are going to accept my proposal. You can play Jedi mind tricks on the reviewer. You can actually force them to score it well for you. One of the tricks that I showed a couple of groups is let's take the impact section as an example. You can absolutely say the successful completion of this project will bring a trifold impact. Just by saying that statement, you already piqued the interest of the review. What is this 3-point impact that they are talking about? So I'm already drawn to it. My curiosity is piqued. Then you speak in bullet point, even though you're not writing in bullet point, speak in bullet point to say that from a scientific perspective, this is the impact that it is going to bring. From a personal perspective, this is how it's going to affect my career. From an institutional ecosystem level, this is how it's going to improve my local environment because I bring this knowledge back. Underline those three factors. The reviewer has no choice but to score it the highest possible rank. What choice do they have? You have just nailed everything. So this is the kind of grand speak that you absolutely have to just force the reviewer into scoring you well. There is no choice, leave them no choice, all right? So this is a very important point. I'm gonna show you an example of my grant that I wrote a long time ago. This is not for an imaging center. This is actually for a research grant. And I want to show you how economically I capture all the essential information in just one single paragraph. And I highlight all the necessary point, right? So what I'm trying to study here is basically cancer cells going through endothelium. It is very important because cancer cell, when they invade, they have to go through endothelium twice. They have to enter the blood vessel, and then they have to get out of the blood vessel. So if you can block that effect, you have twice the chance of blocking something. So my goal is not to develop drugs. This is a real 5-year project, is to basically trying to understand the transendothelial migration process. I'll leave it up to you to read for about a minute. So you notice that I've bolded quite a few important things just to draw the reviewer to the long-term goal of my lab. They immediately know what my lab is doing and what is so specific about the objective of this particular project that I'm applying. And I tell them the central hypothesis, and 3-4 sentences, I get them the preliminary data. And then the specific aims, right? And I'll show you the specific aims later. Let me know if I can scroll. Everybody okay if I scroll? This is how I launch my specific aims before I write it. My 3 specific aims in diagrammatic form. Before I even write my specific aims, I purposely save it to the second page. They already see exactly what I'm trying to pursue. Right? So there are a lot of innovative ways where you can influence the thought process of the reviewer. Anything that will make it easy, usually a diagram, use it. Use it. Even when you're writing for the Army Imaging Center, let's say if you're trying to test 4 or 5 or 6 different conditions, you don't need very fancy diagram. You can just create a table. I have these 5 conditions. These are some of the constructs that will be involved in condition 1, condition 2, condition 3. And reviewers said, okay, that makes sense. The entire work plan makes sense. So don't always have to use Word. Capture this as much as possible through diagram and everything. So that is all I have to say. I'm going to do like a 1-minute wrap up, and then I'm going to pass the mic to Karen and Mike. The reason why I'm doing this, as I mentioned earlier, is the world has changed. We now are seeing dwindling opportunities all over the world for science. And it hit hard everywhere, even including United States. Especially hard are resource limited regions of the world, because there is always the regions that suffer first. That's the reality of life. But opportunities are still there. And this is the reason why I want to do it, because precisely the opportunities keep getting smaller and smaller. If we don't write the most competitive proposal, then there is really nothing much the rest of the world can do to help, because they can't even help themselves at this point. So I hope this is helpful, and good luck with everyone, and feel free to talk to all of us. Like, I cannot mention this enough. Talk to us, talk to us, talk to us. We are really, really trying to help. Thank you very much.
I just wanted to add to conclude not conclude, but to add to what we did today in this workshop. I hope you enjoyed seeing the different perspective, and that it's actually been helpful to understand what goes into the review. So if you're playing the grant game, seeing it from the other side is a huge advantage, right? You know now what it's like trying to understand somebody else's science, somebody else's brain, somebody else's ideas. The exact same things that you struggled with today is what a reviewer struggles with in reality. So I really hope you got to learn a lot from this. It was only a day, very stressful workshop trying to figure out a project from nothing. You guys did fantastically, well done. Leong is not joking when he says that the proposals that you guys generated today rivaled many of those that were received during the first VRP program at AMI. So well done. I think the future is bright, despite what Leong has just said. I think you can't actually, I don't know who said this, but don't forget to take advantage of a crisis. The world is in crisis. That means that we as Africans can actually step up and take advantage of the resources that are available to us. Okay. So I'm going to hand over to Karen, and we're going to kind of wrap up the whole 2-day and the ABIC event.
Cool. Thanks, Mike. I'm going to try find a screen where I have something shared. Give me one moment. Yeah, I just need to find the right desktop. Yeah. Okay, so okay, while Mike tries to solve my hardware issues, I'm going to attempt to wrap up and summarize some key points from yesterday and today, and then talk a little bit about going forward. So the theme across the 2 days, in case it escaped you, and I guess it's also in the title of the meeting, is really highlighting infrastructure, what exists on the continent, what it can look like, what it currently looks like, and finding opportunities to invest in that and opportunities to invest in this community and research on the continent. And we heard speakers from different core facilities, different models, different collaborations and initiatives happening on the continent, the summary of the infrastructure, and amazing opportunities out there from Africa Microscopy Initiative all the way through to the Advanced Imaging Center, which is accessible even though it's international. And a few key points. We heard about the importance of technology needing to support local needs. But remembering that knowledge, I think it was Dr. Karim who said that knowledge extends beyond just histopathology. There is scope for advanced imaging on the continent, and there are sites where that technology already exists. We heard about the amazing equipment already available in Algeria. You heard about Mike. There's really key sites on the continent, but there's also scope to grow that infrastructure in between. Oh. Okay, I'm still trying to wrap up. And there's lots of opportunities in there for leveraging lower tech equipment and frugal microscopy to really try and fill those gaps and build that foundation. We heard about different types of core facility models, free to use, collaborations to use, paying a fee to use, mix of all of the above. And I think what was really key is that we heard that no matter the type of technology or no matter the type of model to access, what was critical was actually the people, and investing in the people and the skills to manage that equipment, use that equipment, train users to use that equipment. And that staffing, those experts take time to train. They do not appear overnight, and retaining that expertise costs money. And I think we had a really, really valuable insight into the reality from two different core facilities, one in South Africa, one in Cote d'Ivoire, the costs of what it takes to run these sorts of core facilities, from maintenance to staffing, and the importance of that institutional support, vendor support, community support, academic institution, and the wider community. We heard about the importance and value of the community to a core facility and an individual, for building those connections, the collaborations we can make, and that's almost more important than the focus just on the science, because that's what enables us to seize the opportunities and do better science. Flipping over. With that, we also heard and acknowledged that there is room to improve a culture of sharing on the continent, but I think the best way to do that is through community. We can be the change we want to see in the world, to quote, I actually don't know who I'm quoting there. And that can start from talking about information. We can share our information and show that how beneficial it is to share our information, to make it easier to access our equipment and our tools, and to advocate for the credit that is due to us when we do that. It's on us to advocate for that. And on the topic of advocacy, we heard about the importance, I think, of telling our stories. It's not just the numbers that get people's attention, although it is a good way to do it, but the context of those numbers and those statistics, whether it's infrastructure, people we've trained, people that we've hosted, papers that we published eventually. The context, I think, is where we get people's attention. And we heard the Jedi mind trick in getting what we want, whether it's a grant or an investment, is getting people's attention and telling them that ongoing investment is what's worthwhile here. So overall, we've heard about the importance of being strategic in what we do. We need to think carefully, because especially in this landscape, we need to make sure that we're building in as much resilience and sustainability into everything that we do. And that fed into today, which you've already had a wrap up about from Mike and Leong, so I won't say much more, except to remind you that it ties into yesterday. It's an opportunity to, whenever you write anything, advocate, tell your story, play the Jedi mind tricks. Your priority is to take the opportunity and secure the resources to build the sustainability that you need and that we all need. Okay, and that brings me to, from a wrap up, looking forward to. You've heard Leong mention that the global funding landscape has changed, and that impacts ABIC as well. We started, for those of you who are not too familiar, we started with a very small online meeting in March 2021. We rapidly, within less than a year, secured funding, and we were incredibly fortunate to do so because the funding landscape was rife for it from CZI. And that grant has funded almost everything that you've seen in terms of building activity, building our online infrastructure, paying a salary for Precious, our coordinator, which makes all of our lives easier. But the key that I want to pull from here is Precious helps coordinate a lot of the all of the actual activity, and she highlighted it, we're all still volunteers. So going forward, when the funds run out in May 2026 or next year, the future is a little bit unknown. But we are actively looking for funding and partners. And I really want to highlight that even if there is a bridge, a gap where we don't necessarily jump straight into the next round of funding, this community doesn't disappear. ABIC's not going anywhere. We started as a volunteer-based organization. We decided with a little bit of naivety, hubris, and enthusiasm that we could try and do something on the continent, and we will be able to keep doing that, right? We've not been sitting passively. I would just want to highlight, we have been engaging in the background in quite a lot of strategic planning. One of the things that we spent a lot of time with was the stakeholder engagements with global communities, funders, key partners such as Leong, global bioimaging, even vendors. And I just want to highlight some strengths that our partners identified for us, because I wanted us to take encouragement from this and to think about what we can do to leverage that. Everyone identified that our strength is our community. We are connected, we're open and inclusive, we're vibrant and we're collaborative. We're a very active community. We've been strategic and we are being strategic about what we do. And our strong community base lends us an incredible legitimacy on the continent to ourselves and to our institutions and to funders and vendors. From that, we have a whole bunch of opportunities. We're still going through the strategic planning process at the moment, so I'm not ending with any grand announcements of what we're going to do next. I do want to highlight, though, that out of a lot of these opportunities, you will probably see us shift to focusing on what we can do across our digital platforms, because we're running out of the funding to meet in person without a lot of fundraising, and then focusing on what we can do. I think this is a great opportunity too. Over the past, especially past 2 years, we've seen from ABIC, SABI, South Africa Bioimaging, has grown in strength and activity. We've had an emergence of Nigeria Bioimaging. This year, we've had an emergence of Kenya Bioimaging, which might become an East Africa Bioimaging. There might be, I hope, an Egyptian Bioimaging coming soon. This means that we have the opportunity to partner with regional networks where you, in your regions, can focus on what can you do, how can you interact with your institutions, your different vendors, partners, etc. And it will distribute some of the activity and coordination as well. And then we've got some clear ideas on how to scale some of the specific training needs that we know about the continent, are on the continent, and how to leverage the infrastructure that already exists. This is very high level to just say, if you see ABIC, or what you see coming out of ABIC is going to change probably over the next months to years. Normally, I would stand up here and announce where the next meeting is going to be. There is no announcement this time because we don't have the funds to promise that we can do this in person next year. But there have been some conversations today. We have a plan. It just might not be next year. So please watch out for announcements. Watch out to see what's coming next. But critically, as I said, this is the community. Those of us in this room, those of us online, even those of us who couldn't make it. And this carries on by the community seizing opportunities, getting involved, getting engaged, staying engaged, bringing your ideas to what we can do for this. And we really look forward to partnering with you all. We're going to continue. We are resilient. The landscape will change again, it always does. And I think the best is yet to come. So thank you, everyone. And I'm going to hand over to I'm handing over to Mai now to do the last bit of the wrap up.
Oh, so this moment is always a sad and a very, very proud moment that there's that we did the ABIC annual community meeting. This year, as I said at the start of this meeting, is very special to me because it's in Cairo. So this means that ABIC is growing stronger and stronger. We're connecting more countries on this continent. So this is a good thing. And actually, as said, as Karen were saying, like a lot of things are coming better, although we're looking for funding, but still as a community, we're growing. And this is a really good thing. I want to thank every participant for your sharing their ideas, sharing your questions and challenges. This is really what ABIC needs to know, like its direction. And I want to thank all the organizing team, Leon, Karen, Mike, Precious, Hadil and Khalid, and also for Dr. Ahmed and Dr. Karim for hosting us at the American University in Cairo. Thank you so much, and we're done.